Hi everyone, this is the second video that you should watch for Thursday, March 19th. And the goal of this video is that you will understand a bit more about genetic mapping. And that will be important because you'll be writing about mapping for your research paper. You can consult in the red box below on this first slide all of the things that you'll be wanting to work, all of the things that you should be working on to get ready for next Tuesday, March 24th. Okay, so why is genetic mapping so important, and why are we going to spend some time talking about this and giving you background? Well, the paper we're going to talk about, the first paper we're going to talk about next week, is on how they mapped the, or made progress towards mapping the gene for cystic fibrosis. And in the big picture, we need to first map human disease genes in order to eventually clone the genes. Mapping means that we are figuring out which chromosome the gene is located on and on which part of the chromosome. So eventually, by doing mapping, we can identify genes that are associated with the disease. By mapping genes to chromosomes and regions of chromosomes, this is really the first step towards eventually being able to design therapies. Right? We need to map the genes to chromosomes to allow us to clone the genes, and once we know the sequence of the gene, we can then start thinking about understanding how that gene functions and how we can help individuals whose genes are not functioning in the right way. So the material that this PowerPoint comes from is the chapter by Thompson and Thompson. It's an online textbook called Genetics in Medicine, and this is freely available through the CMU library. The link may work for you directly, but if it doesn't, you can search for Identifying the Genetic Basis for Human Disease in the CMU library search bar. Okay, that really is going to be optional in terms of accessing that. You don't absolutely need to read that chapter, but you could use it as a supplement to the PowerPoint if it's helpful. So there are three basic approaches that Chapter 10 describes, and the first one is linkage analysis. Linkage analysis is what we're going to talk about today. It's a family-based approach to map genes. That means we're using pedigrees, and this is most commonly used for single gene or monogenic disorders. A second major approach is association analysis, or GWAS. I mentioned this one briefly in the March 17th video. This is more of a population approach where you have non-Mendelian inheritance patterns, so you can't really use pedigrees in the same way. So for complex diseases, this is one of the ways where human disease genes are mapped. And the third approach is direct genome sequencing, and this is really fairly newly possible with high-throughput DNA sequencing. Okay, so we're going to move on to the second point here and talk about 5 different aspects about genetic markers that are used to map disease genes. And the first thing we're going to do is just review recombination and what is a genetic map. So, for example, this would be a genetic map for Drosophila. Drosophila, there are only 4 chromosomes. So you can see that on chromosome 1 on the far left, yellow body is at the far tip of chromosome 1, and located nearby is scute bristles and then white eyes. So those 3 traits are quite close together, and they would probably tend to travel together in terms of being passed on as alleles from parent to offspring. In contrast, you can see at the other end of chromosome 1 is bobbed hairs. That's located very far away, and therefore it would tend to actually probably assort independently of those traits, the yellow body, scute bristles, right? It's very far away, and so it's not likely to travel together. So by doing either 2 or 3 point mapping, you can actually determine the location of genes on chromosomes. And again, we need to get this kind of data in order to eventually clone and sequence genes, and then to figure out how those genes function. We have seen an example of gene mapping that has led to gene cloning in Module 2 in the C. elegans program cell death paper by Conrad and Horvitz. In Figure 1, we saw that they mapped the EGL1 gene to a chromosomal location, and then they cloned the gene by testing small fragments. So in the text, I've highlighted where they mention this, and this is shown in Figure 1. You may recall that this is chromosome 5, that LGV, stands for linkage group 5, and a linkage group is the same as a chromosome. And they mapped the gene because they were working in worms. They could set up multiple crosses, and they figured out that EGL1 traveled with ROL4 and UNC76 a lot of the time. So therefore, they were able to place EGL1 on the chromosome, chromosome 5, in between ROL4 and UNC76. Now, you might wonder, well, we can't really take the same approach in humans. We can't actually set crosses and ask different human individuals to mate together to see what traits travel together. So our approach for mapping genes in humans is going to be a little bit different. The next step in the Conrad and Horvitz paper, after they mapped the gene and narrowed down the location, they were able to test DNA fragments in that location by injecting them into worms. That was the transformation rescue assay they did. And again, that allowed them to clone the gene eventually and get the sequence of EGL1. Again, we can't do this in humans because we're not going to be testing the right DNA fragment by injecting DNA fragments into any kind of human. So the approach to validate the genes is going to be a little bit different. But conceptually, we have the same process, mapping leading to cloning. Okay, so mapping human disease genes, if we can't set crosses, it means we're going to need to use genetic markers. So the goal for human disease genes is a map that shows the order and distance apart of all disease genes. And clearly, we don't have this quite yet. But we know that the map distance will be equal to the percent of recombinants. So the reason we don't have this entirely is that disease mapping is not usually feasible in humans. It would require double heterozygotes, so people who are heterozygous for two different diseases, and then we would be able to do this kind of mapping. But again, very rare that people would be heterozygous for two different diseases. When we look at pedigrees, there's going to be another limitation, which is that only sometimes do they reveal these recombination events. It depends on what kind of information is available for all of the individuals within a pedigree. We would need to know that a parent is heterozygous, and then we would need to know how the alleles are arranged on the chromosome. So this would require DNA sequencing in order to get this kind of information and be able to know that recombinants have happened and that individual offspring are in fact recombinant. Okay, so what genetic markers are used in mapping human genetic diseases if we can't do disease mapping? Well, they can be really almost any Mendelian character, and what that just means is any piece of DNA. But we want to use markers that are easy to use and that are inexpensive to score. So the criteria people have used over the past decades is that first, the character has to be polymorphic. And what that just means is it has to vary. A randomly selected person should have a really good chance of being heterozygous. Okay. And a breakthrough in terms of finding genetic markers has been finding them spaced throughout the genome. These would be markers such as VNTRs, variable number of tandem repeats. And on the next slide, I'll show you several of these different kinds of markers that have been identified over the years. So these variable number of tandem repeats, this is the large part of the genome. Remember, over 50% of the genome is composed of repetitive DNA. Only 1 to 2% is protein encoding genes. So within this repetitive DNA, there is a lot of variability because there's no pressure to keep that DNA the same, right? It's not carrying out an absolutely important function. So there is a lot of variability, and therefore we can look at that repetitive DNA and compare it from one person to the next. So when the Human Genome Project was being worked on, more than 10,000 highly polymorphic markers were generated and mapped. Okay, so you can kind of see like the history of what markers have been used. Blood groups were used at the very beginning of the 1900s, and you can kind of see that we've kind of moved on to more of a DNA approach, and those VNTRs are here sort of in the middle. They were called mini satellites. That was the mid-1980s. And they were highly informative, and there were many alleles, but they tended to cluster near the ends of chromosomes, and you had to figure out what type people had by process called southern blotting, which was not necessarily the fastest approach. As time goes on, you can see in the late 1980s, people moved onto microsatellites, which are smaller repeats, either di or tri or tetranucleotide repeats like CG, CG, CG. And you can actually type those by automated PCR, so that's much faster. Oh, sorry, I went forward when I did not mean to go forward. Let's see if we can go back. Oh, doesn't seem we can go back. Okay, well, that's all right. So the last type of repetitive DNA that's been used are SNPs, single nucleotide polymorphisms, and that's really what's used mostly today. So once we know what these different genetic markers are, we can talk about two-point mapping and LOD scores. So how can you determine linkage between a disease and an informative marker? So what you're going to do is use a mathematical formula that I'll show you in the next couple of slides, and you won't actually have to do the math, but this is what researchers do. They are going to determine a recombination fraction, and that's called theta. So that's that symbol right there. And that's one of the questions that's going to be asked on assignment 11. So you'll be able to refer back to this PowerPoint to help get some of these answers. And once you know what theta is, you can use a statistical test to see if the recombination fraction is significantly different from 0.5. And 0.5 is the value you would see if there was no linkage. Okay, so if we take a look at that family that we had looked at previously, we can see how we can calculate the recombination fraction. So in this pedigree, we were able to identify recombinants, so there was enough information, and we can calculate theta because it's equal to R, the number of recombinants, divided by the non-recombinants plus the recombinants. So in generation 3, we have 2 recombinants of a total of 7 individuals. And so 2 recombinants in these 7 different meioses, right, in the 7 different offspring, means that theta equals 0.28. So that is how you calculate theta. Once you have theta, you can calculate the LOD score. Okay, so you can see in the gray box is where the actual formula is, and again, you will not need to use that formula. But it's helpful to kind of understand that theta is an important part of calculating a LOD score. A LOD score is the logarithm of the odds of linkage, where the odds of linkage is the ratio of the likelihood of linkage and non-linkage. So it's a logarithmic score. So what that means is a LOD score of 3 plus 3 indicates 1,000 to 1 odds that the linkage being observed did not occur by chance. So what that means is that's a pretty good LOD score, pretty high odds that the marker and the disease are really truly linked. Okay, so basically what researchers do is they test many, many, many different markers with their disease, and they're trying to find a marker for which the LOD score is high, right? So that is the basic idea. They can get complicated pretty quickly. Software can be used, but there are limitations of the software. And as I was saying, the second point on this slide is that families have to be typed for many, 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 many markers until those positive LOD scores are found. And this is often referred to as sort of like the needle in the haystack kind of approach. But when you see high LOD scores, some people use a threshold of +3, others would say +5, but this is going to be a strong indication that you are close to where the disease gene is located on a chromosome. In contrast, if you have negative LOD scores, that's going to help you exclude regions of the chromosome, and it tells you that the gene is not in that area of the chromosome. So even though a negative score is a little bit disappointing, no doubt, it is actually still informative to help you narrow down where on the chromosome is the gene. So in reality, researchers often try to analyze more than just 2 loci at a time, just to try to be more efficient with linkage analysis. And I'm not going to say much more about this whole idea of multi-point linkage, just to tell you that there are limitations. Some of the limitations can be the small size of families and missing information on individuals. If you can't say for sure which are recombinants, that's going to be very limiting. And so instead of doing marker disease mapping, sometimes individuals will do, or researchers will do marker-marker mapping in order to overcome this limitation. And what you don't need for marker-marker mapping are large numbers of families with the disease. Okay, so that really is a huge a huge issue, especially for autosomal recessive diseases that can be quite rare. So this has been really successful. High density maps have been accomplished by 1998, so going back now many years. They used just 8 ideal families that had really good structure, and lots of sequence information was known, and they used 8,325 microsatellites, and that resulted in over 1 million different genotypes. So basically they were able to with precision, figure out where these different markers were located. Okay, so if you have some good pedigrees and we know a lot of good marker information, it seems like maybe this would be, putting us in a really good position to map genes and clone human disease genes. But there are still yet more limitations for LOD score analysis. Other problems that can occur is sometimes paternity is not accurate within a family structure, or the diagnosis of the disease may not be accurate. There's also computational limits, and there's also something called locus heterogeneity. So that's a situation where hetero means multiple or different genes. So heterogeneity is different genes are giving rise to the same clinical phenotype, right? So that would be the opposite of monogenic, if that makes sense, right? So locus heterogeneity means mutations in multiple or several unlinked or different genes are giving the same phenotype. So that's more of a complex disease situation, and LOD score analysis does not work well for complex diseases. So basically, you need to specify a specific genetic model for each disease. You need to try to figure out the mode of inheritance, which is why we talked about pedigrees. You need to try to get a handle on how penetrant the disease is, so you know how accurate the phenotypes are for the family members. And again, these mapping methods are not going to be applied for the common complex diseases. But you will see when you're reading papers on any of the monogenic diseases, you will see that people use this pedigree LOD score approach to try to map the disease gene. Okay, so I'm just going to define a couple more terms to get you ready for assignment 11 for next week. Linkage disequilibrium is a term that you'll see in these mapping papers. This refers to allelic association between genes. And basically, this just means that genes are linked, that genes are not moving independent of one another, that they're closely placed together on the chromosome. Okay, so it's kind of an odd term, I think. You can probably safely think about it just as linkage, but it has this longer term linkage disequilibrium. Okay, so genotypes for markers across the candidate region are going to be analyzed in unrelated affected patients. And if they have independent mutations, well, then you're not going to learn very much. And that often will happen for dominant diseases or X-linked diseases. But if you are fortunate, if some of the genes in apparently unrelated patients come from a common ancestor, and that often happens with recessive conditions, then a shared ancestral haplotype may be defined. And this was the case in the path of cloning the cystic fibrosis gene. So again, here's another term that crops up, haplotype, and that's just a set of DNA variations that tends to be inherited together because they're close together on a chromosome. So a haplotype would be a combination of gene X and gene Y, because gene X and gene Y are located close together on a chromosome. Okay, so what allele are you for gene X and what allele are you for gene Y? If you're going to inherit one, you're probably going to inherit both. Okay, so linkage disequilibrium and haplotypes, those are terms you're going to see in the mapping papers. And the last term to define for today is autozygosity, and this is the idea of using inbred families to try to map human disease genes. So autozygosity means homozygosity for markers that are inherited from a recent common ancestor. So an example would be if parents are second cousins, they're going to share 1/32 of all genes, and the child will be autozygous at 1/64 of all loci. So these small inbred families that do occur in certain communities can generate significant LOD scores and can be a very powerful tool for linkage analysis. There are examples where such families have contributed to researchers being successful in mapping and cloning genes, for example, hearing loss and benign recurrent intrahepatic cholestasis, as well as cystic fibrosis. So in the paper that we'll be looking at next week by Watkins, you will see some pedigrees of these small inbred families. Looking ahead to next week, I've mentioned the Watkins paper a few times. So, the assignment number is missing there. It's assignment number 11. It is listed under the Module 3 assignments in Blackboard. And I'm asking you to go through the Watkins paper, which is quite short, and it's just a series of questions to kind of lead you through the paper to try to see if we can understand the big picture of the Watkins paper. And again, remember to use those discussion boards to help one another as you like. Again, these will be graded on your best and complete effort. There will be a new PowerPoint video posted on how human disease genes are cloned that you can watch as well for next week. And you should hopefully get started working on your draft of your research paper, parts 1 to 3, as soon as possible. Thanks, and have a good day.